Okay, so our speaker has been you. Just hand it over to you. And you is here at the statistics department.
Well, thank you for the organizers, Fritz and Terry, for having me. So as Fritz said, I'm a statistician and also have appointment in EECS and also computation biology. So my perspective is more somebody coming from outside. I have been working with the Galan Lab for the last 10 years, so more like outsider view, but I do have a collaborator with Jack. And so share some of our thoughts. And also a lot of the experience also taken from other data science project in other fields. So I see it a lot of statistics is the field that you transport a lot of good ideas from field to field. And I really welcome comments during the talk. So now a lot of things are being put under the umbrella of data science. And I think it's, for most of us, easy to agree that there's three important components: computer science, math and stats, and also domain knowledge. Here is neuroscience. And I just want to remind us that actually early development computing devices were partly driven by data analysis. 1890, Hallowitz, a statistician at Census Bureau, faced the problem of processing data, which was predicted to take 13 years. So US Census every 10 years, so that was not desirable. Therefore, he was charged to develop computing methods or devices to speed it up. And he developed Hallowitz tabulating machine and had a company that merged to IBM as one of the four companies. So this is a history that data have always, large amounts of data relative to the time, also driven computing development, and Fisher was really a geneticist. And the Berkeley definition of data science is really where we're merging statistical and computational thinking. And machine learning has been very, very active field of both statistics and machine learning, and both computer science and statistics, and prediction and cross-validation both, were there in statistics, but never at the center. And the first generation of machine learning, I would say, that really built on prediction optimization and made a heavy use of cross-validation. So here we really want to do science. So I'd like to see cross-validation as actually application of very scientific principle, right? Prediction is a big principle for science. It's, you have a discovery, you have to be replicated by another lab. And in a lot of the astronomy developments, like Gauss and Kepler, it's really about prediction of stars, right? So it's about one validation of scientific theory is predict the future and accurately. And I see cross-validation is really try to use the same data under consideration from science and data collection to say that actually we can think of having 4 experiments, and we can delete one part, and then use the 3 parts to predict, and we can cycle through. So it's really like a pseudo-replicates idea. And the second principle of data, like science and data science, is really replication. And as I said, right, machine learning really part of statistics and machine learning and computer science. It's really making huge strides in all kinds of areas. Prediction is very, very easy to describe and understand. And you have data availability, computing software availability. So it's really all you hear about is about machine learning. But I think machine learning and statistics are kind of coming back together very nicely in the sense that we really now care about interpretation. EU had a regulation basically give users rights to explanations. And now you always say statistics, we like smaller models, and we want to really interpret in a way that between scientists we can communicate and also be much, much broader audience, right? Easily you can see precision medicine, we're going to have treatment based on machine learning statistical methods, and you want to tell the patient why this is a good one instead of, oh, I'm a good doctor, and therefore you should trust me. Why should we trust the algorithm? And data science, what I've talked about, the three principles a lot, is based on research and also teaching experience. We have been struggling to say that how can we make statistics and data science accessible to a much bigger audience. So I ran this statistics class 8 out of 10 years, PhD level statistics called 215A in Berkeley. And 2/3 of my students are outside PhD students. Right? They're not really just for
How many?
Last semester I had 40. So it's because we have essay type of reports, and we like to scale it up, right? So that's where we are at. And one is like they don't have time to take 2 years of statistics classes, and many of them have done MOOC on their own. And how can we describe or organize statistical knowledge in a way it's more like physics principles, and so they can get quite a lot out by knowing the high significant bits and go to detail instead of they have low everything before they can do anything. So that's a lot of my teaching thinking merged into practice. I also like to be able to enlarge the umbrella of probability thinking to include like PDEs and things not exactly probabilistic, right? Applied mathematicians are very much doing data science. So that's another thinking for these three principles, go beyond probabilistic modeling. Another thing we follow very closely in my lab is that we actually develop general methodology. That's basically our task as statisticians within the context. And we try to solve problems before we say this is the general methodology other people should use. So I take that very seriously, that empirical evidence for something to work instead of just some simulations and seem to work. And so I summarize, I give you this is really going the other way around. I have been working on this for the last 4 or 5 years. Summarize as called PCS workflow. So a lot of people I think already doing this, the top labs, and but we now want a lot more people take advantage of what I would say the top labs doing, so that they don't have to take as much time to get the same gain. So it's really building on machine learning. Use prediction as a form of reality check, right? In classical statistics, we have goodness of fit, but in high dimensions, kind of have to do, and we don't talk about it anymore. And that's very dangerous. I have definitely seen in qualified exams, people choosing two horrible hypotheses. But the framework doesn't stop them from doing that because they're two hypotheses. So that's one thing, is that we need to have some way of measuring the model you develop, somehow capture reality. It doesn't have to be prediction, but prediction is an easy one. To say that it fits. What do you mean by fits? And the measure usually comes from the subject matter. That I would say that in the first example is really measured against the experience. And computation, actually, I don't talk too much about because my lab actually do computation, but I think our strength is actually both on the theory and the scientific collaboration. And stability is something I've been advocating as a minimum requirement for reproducibility and prediction. And recently, actually interacting with Bruno and Fritz, we also see it in neuroscience. Yes.
So actually, it's interesting that you leave out computation because that's what most of us do.
Yeah.
But I'll tell you, the key principle in computation is scaling. So when someone comes up with an algorithm, and there are a lot of algorithms out there, you should be asking yourself, well, not just whether it does what it claims to do, but how well will it scale up if I increase it by a factor of 1,000 or 1,000,000? Very few algorithms scale well. From AI, none of them scale well. And the reason why you've been hearing about deep learning is that it's the only one that's scaled to the point now we're taking on real world problems. So it's computability, not computation, but computability, whether you can actually compute it with current hardware.
Yeah, I agree. But
by the way, we really want this to be interactive. So if there's, I'm interrupting, not just to fill in something, but also, from the point of view of interactivity, we want these lectures to be interactive.
But one thing I want to push back on that is, I agree that.
Yeah. So is it the algorithms that are the problem in terms of scalability, or the hardware?
Both. Actually, you're right. If the hardware hadn't scaled up by a factor of 1,000,000 over the last 30 years, no algorithm could have been simulated. But it's a combination of both. And what's happening now is very interesting. You mentioned software for machine learning. So it's now, there are packages out there like TensorFlow, which run on special purpose chips, which give you, many orders of magnitude of speed up over traditional computers, because these are parallel chips that are able to take problems that can be parallelized, and deep learning can be, and makes it much more efficient. So there's billions of dollars being put into the hardware as well as the software.
So I agree that computability is extremely important, but I also want to push back on that. Actually, that's mostly talked about. So I want to emphasize actually interpretability and stability. You can compute whatever you want. Actually, we deal with it, but it's just not emphasis to talk. But the result is equally important, that it makes sense, and also we can validate it. And I actually feel like it's less emphasized. I definitely agree with you than computability in usual talk. So that's what this talk more is about that. So we're also writing a textbook with my student Rebecca, and writing a paper on that. It's really seeing stability and computability. Computers, like, it's the trunk, right? It's very, very important to support all the fruits of the tree of data discovery, and stability is also very important to ground it. Definitely, I agree that computability is hugely important.
Sorry, what do you mean by stability?
I will define. Yeah, thank you. So stability, exactly right, seems make sense and then self-evident. As a philosopher friend from Northeastern, he gave me this quote from Plato, right? That's why knowledge is priced higher than correct opinion, or knowledge and differs from correct opinion in being tied down. So this is just self-evident that knowledge, even words, the meaning have to be stable. And we came to this, actually working with Jack's lab, we were doing lasso, different predictive modeling, and we want to interpret. And then if you change the data by 10%, I thought they could decide to only spend 10 minutes less to record the data. You don't want the important predictor to change. So actually that's the beginning of this thinking about stability. And now, of course, there's a literature on robust statistics, and in many fields have the stability concepts. So I'm slowly putting together as a principle. So that's going backwards. So what's stability I mean? So I started with thinking that uncertainty and sample stability, right? You subsample in a reasonable way. And then I'm like really extending it to beyond sample-to-sample variability to the whole pipeline of data science, from pre-processing, right? You want your pre-processing method not to be determining what your outcome, right? So a lot of the data, as Terry said, is pretty complex. More and more, the process, unlike the single neuron measurement, it's pretty long with unknown. A lot of people actually don't know how the data is processed. And in bioinformatics, definitely the normalization for omics is very popular. And there are different ways of doing it. How would you know your result is not an artifact of particular processing? So that's right there is stability. And also data visualization. You don't want to look at the pattern and then think there's something there when you change the data a little bit and the pattern disappears. So it's robustness in its most general form. And also human decision, this paper from Liam's group, Fritz pointed me to, about high throughput imaging data in neuroscience. They made many choices on the parameters in the way to going, right? They do multi-scale, it's about experimental design, but they had a lot of ad hoc choices human made in coming up with the conclusion and the processing. And we want to make sure that's not dominating the results. So we do many, many things we don't report. We say, oh, we just set this parameter or set that parameter. Before, it's less a choice, but now for deep learning like TensorFlow, you have architecture, you have many things people can tweak. And the worry is that even you can compute, but if you tweak too much, you're overfitting.
And
the
hyperparameters.
Exactly. And also target of interest. Stability has to be defined relative to a particular target. Things can be stable for prediction. That's what deep nets is, but not to interpret one of a few million weights. That's not stable, right? And also, we have to be transparent with how we decide on these metrics. Is, of course, technical knowledge, but a lot of experience and judgment, and even regulation will come into play. So all of that, I like to see a more transparent workflow reporting to really have them all nicely boxed and described in a very clear way so people can decide on the evidence you present.
So question, how will you talk about how this can be realized? Because it appears to me this is really also, it's a data management problem. You need to really, in a very transparent fashion, be able to actually document in a data set how actually the pipeline is configured, what the hyperparameters are, and things like that.
Yeah, there's a small group of people, statisticians, have been meeting to use Jupyter and IPython platform to record. And in the future, we can even use a machine learning algorithm to mine. So we can go into all the recording, the logs, to know whether you have touched the data how many times, right? So technology is more there now to make people more.
So by the way, this is a really important problem for experimentalists, because it's not enough just to collect the data. You have to specify how you collected it. What were the conditions of the animal? What was the the protocol? And if you don't have that in the data set, then the data becomes much less valuable, because maybe that some of those parameters, like the temperature or the time of day, may be critically important. And this is why often things can't be replicated in different labs, because they don't actually, all the information that you need to reproduce experiment isn't in the paper that's published.
Yes, exactly.
And so this is going to be critically important. All of this other metadata is equally important as the actual recording.
Yeah, I think the community we can agree on a certain way of reporting that. Just have boxes people have to fill in so they can now avoid it, right? Good labs always do that, but not all. Even they record it in their own lab book, they might not put in the paper. Yes?
In your slide, what does the EDA stand for?
Oh, exploratory data analysis. Yeah, thank you. Yeah. So it's like visualization. Yeah. But I think that's one community can agree on, like a journal can say this is the reporting protocol, and you just have to fill it out on GitHub or something if you don't have space in the paper. So this is just a quick slide that this is really unifying a lot of papers in the literature in various forms, and I won't go into details there. And just say that this robustness stability is extremely fundamental together with predictability, right? It's like we do so many things, just, let's try this, let's try that. And then I'm really worried that in the end, you're basically reading the noise. And I also want to move away conceptually from, you always have to have a true model. A lot is approximations. I think philosophically it's better to think about all models as approximations than, they are generative. It could be generative, but you have to make a case for it. Then just automatically say that, because probabilistic, it has to be generative. Yes?
Just a couple of questions. So one is, it's unclear to me, so you started out with these 3 different domains, right? Like predictability, computability, and stability.
Right.
But I guess from a non-statistician's point of view, as you also noted here, stability is essential for good predictability, at least in a cross-validation setting, right? Because your validation, because your data sets are slightly different in cross-validation, right? You've left out some data. It can be viewed as a form of perturbation.
Well, I would say that they do overlap. I do agree they overlap. If your target is prediction, stability and predictability very much aligned.
Yes.
But I'm trying to give a possibility that stability is not about the target you predict, it's about the parameters. So then they separate. Okay. That's a very good question.
Okay,
yeah.
All right. And then the other one was exactly the point you just made, which is that, I guess I'd just like to resonate with it, which is that indeed, right, in the real world, when we're modeling data that's thrown at, that the world is generating, I think the chance is like measure 0 that we would ever hit upon the true generative model, right? So in fact, a lot of the fallacies that we encounter in applying some of these statistical measures like BIC, et cetera, assume that one of those models is the true generative model, and really neither of them is, and so then we need something to deal with.
Yeah, that's definitely I agree with, but I'm really trying to say something deeper. So when you even define a random variable, you need to think about the physical process to generate the randomness, and we usually, especially teaching, we gloss that over very, very quickly, right? Everybody feel like, I used to teach for 20-some years, statistics start with x1 through xn RID, right? And I just now is horrified myself. That's how I did it, but that's how I learned it. But I've tried to move away from that and introduce random variable very, very carefully, like through I don't introduce random variable until mid-semester now when I teach.
So this is something that, again, everybody makes assumptions, and if statisticians make lots of, one of the assumptions they like to make is, let the noise be Gaussian, right? Or
no, we move it away from that.
In other words, no, they have. And so this is something. When I was trained as a theoretical physicist, I had every day I prayed to the God of physics let the world
Be Gaussian.
let the noise be Gaussian in the world. Let the equations be separable, those are the only conditions under which you can actually come up with analytic solutions or theorems, right, in statistics. Now we're living in a world where, in the brain, any images, they're not Gaussian. Data from the brain are not Gaussian, and the equations are not separable. And so we really have to deal with the real world. That's why it's really, even from the statistical point of view, it's a tough problem.
Yeah, we should just be very, very, either we should make this assumption, actually we don't have any real evidence, for whatever reason I have to do it. Just say it very clearly, instead of muddle it up, right? What's real, what's not, right? We all make assumptions, but sometimes I think always it's good to say what's really supported by experience, or the empirical data, which is like for theoretical reasons we have to deal with it because otherwise we cannot compute. So be just very, very transparent on that. In terms of data perturbation, right, we mentioned cross-validation. But clearly, you need to have, when I do this blocking, for some problems, for time series, you don't want to do that. You don't want to take a block out. A lot of the neuroscience data actually is time series dynamic. So you might want to do that, but you need to make an argument why that's a good idea, why that's like a replicate of your original data. So all these different forms also have assumptions. And one thing I want to point out is that for the data perturbation, except assessing kind of sample variability and all of that, that's the first bullets. It's also emerging from the deep learning literature that you can also use data perturbation to enforce stability, like the adversary examples in deep learning, right? You actually strategically add certain examples. You don't have to change your algorithm. So this is very much a data and algorithm-centric view, that I don't want to change my algorithm, I change my data. So I can have the whole computability of computing structure intact, but again, in terms of regularization. You can do penalization, but then you have to change your algorithm. But this way, through the data perturbation point of view, you can keep your algorithm intact, but change the data. So that's from computability point of view, it's very desirable. So you didn't waste your time, build this huge computing structure, and now you do a different regularization. Of course, you can modularize things, but this way you don't have to change anything except changing the data. So that's a good idea in this computing and data-centric world. And model perturbation, that's what I'll show you, that I have 18 models, give equally good prediction. Then what do you do, right? Before, everybody has their data tagged away, and they did one analysis, nobody can touch it anyway, so we have to trust it. But now, if data is public, as said, any people can try. You can end up with lots of different results. How do we consolidate these different discoveries? So I said, let's do stability. Let's take the part that we all agree. For the part we don't agree, we better say why this model should be trusted more than the other one, or this algorithm. And otherwise, we take a conservative approach. We're not in a phase that we have too few discoveries. We're at a phase of too many discoveries. So it's better to be conservative. I have a problem even with machine learning and statistics papers to build on other people's work, because I just feel the evidence is not enough. And then we're all lesser off because we cannot build on each other's work. So that's more like proliferation of different algorithms. So about causality, right? In this game, we're in science. We try to do science. So we have Hooke's law, or Kepler's law, that's kind of the holy grail of causality. Even though we never really can change the intervention in Kepler's case, but we all trust it. But for Hooke's law, we can do experiment, do intervention, and see things happen. On the other hand, you have statistical causality, or FDA, for drug testing. We want to see the average change for a new drug and things like that. And precision medicine is coming towards individualized treatment, but you can never go to one person. You also have to have a small group. So coming back to science, prediction we know can be just association, doesn't mean it's causal. But if you add stability, I think it's a stronger evidence because you get interpretability and you can go for design for the next experiment. So that's also for stimuli generation, so that's the first. So for the rest of the talk, I will have to go. So I will show you kind of old technology. I'll go back to 1960 single neuron measurement because the access stability problem with real data. I mean, we don't have multi-neuron data. We cannot access because Jack has some, we haven't put our hands on it yet. So that's the problem I face. I cannot access the modern data because I'm not inside neuroscience. But we try to bring to the project actually pretty contemporary machine learning statistical tools. So that's what I'll see. So unfortunately, I don't have multi-neuron data to show you. And the second one is on genomics, but you can also use this technology to analyze genomics data related to neuroscience. And non-extreme matrix factorization and stability comes back for selecting number of components. And I'll try to finish with some lessons learned. I was at a DOE workshop, and they taught the word called scientific machine learning I quite like. So I borrowed here. It's basically use machine learning to do science. So this is the first project. This has been in the work with Jack's lab. My group is leading it for the last 4 years now. And Ben Wilmot collected the data now about 10 years ago. So the data, nobody has published anything like that. 2 leads, Riza and Yuansi. Yuansi actually here, and Adam and Mike and Ben. So we're looking at the V4 area. For this group, I don't think I have to explain what's V4.
No, you should,
You
because
should.
there are people here
Okay.
who don't know any Vs.
Okay. So this is one of the 2 visual information pathways. One, there are 2, one is what, one is where. So this is the what pathway. You see V1 is more Gabor edge detection, V2 kind of similar, and you go to V4. V4 has been very elusive to analyze because it's not object recognition, because that's IT, but it's seem to recognize more complex shapes. So people have made these synthetic shapes and to try to probe the neurons, right? So that's limited by our imagination, how you make out this. And complex can be infinite different ways. This is just what people have done. And this is another note. People know that using this curvature synthetic stimuli, know that some V4 neuron care about curvature. But this is very, very controlled, and it's not natural at all. Right.
So these are preferred stimuli for single cells in V4, right?
Yes. Yes. This is you can see different cells. It's not good resolution.
Yeah,
prefer certain shapes.
So just a background for the non-neuroscientists. So when Hubel and Wiesel first put their electrode in, they made a discovery that eventually led to the Nobel Prize, that unlike in the retina where the cells respond to a spot of light, in V1, primary visual cortex, they responded to oriented bars and edges of different orientations and locations. And so when you go up from V1 up to V2 to V4, the cells respond to more complex stimuli. And now these are attempts to come up with a basis set and see whether which of those the cell responds to best.
Yeah, so that traditionally the neuroscientists have been characterizing single neurons by what stimuli excite this neuron. So that's the same tradition we'll take. So we're going to use deep learning, try to find those stimuli. And we hope you agree that it's a lot more natural looking than this. So this data was collected, 71 neurons from awake macaques collected by Gallant lab, and black and white because it's done quite years ago. And so randomly selected images being fed to to understand a particular neuron, and it's decided by some pre-experiment result to know that it's V4, right? So that's the Jack lab decided that. So related, we had the early analysis still being written for 5 years now, that for 2, we can get the state-of-the-art prediction performance. What I mean by state-of-the-art is compared with V2. So we have V2 prediction results at certain prediction accuracy, and we use that as a benchmark to understand V4. So we claim a success if we match that. So that's what we mean by good prediction. And we use 2-level sparse coding, got pretty simple relative model. And before we did the work I'll present, and James DiCarlo's lab has been a parallel independent line. And their image is a little different. That is semi-natural. They have objects imposed on natural stimuli, but they did very similar prediction work. So we have been holding it up and try to do interpretation work. But we think our work actually is not wasted because we replicate. It's a different lab, replicate the results. So it's a good thing. But for publication, usually you have a harder time. So we're pushing really hard on interpretation. Right? But I want to emphasize it's good to have our lab replicate what James' lab did independently. We started with the second question. What do V4 neurons do? Right? We're looking for stimuli that make neurons excited. Indirectly, we learned some evidence to whether convolutional networks actually resemble brain function. Now, we were surprised because we started with our own deep nets, and I was very surprised when Yuan-Sen research showed me that we can do pretty well with deep learning. So it's transfer learning, I'll explain, and we have the state of prediction, and we go for stable interpretability over 18 models among good predictive models. So ImageNet is this machine learning computer vision database of about 14 million labeled images into about 1,000 categories, cat, dogs, and things like that. And it's color images, and this is a particular architecture developed by AlexNet Hinton's group in Toronto. We just took it, and we said if we use that as black box to process our black and white image as a feature correction, we do something very simple, regression with regularization. Can we predict the average response rate of the V4 neuron? So one of them is the up and down is particular neuron response. Yes?
Are these categories provided by humans?
Yes.
So humans have pre-classified these.
That's the training data, exactly. So that's why.
So that might introduce a bias, right? So V4 might be looking for something that.
Exactly, but amazingly work really well because we take the early layers. So that was a nice surprise, actually. We didn't expect to work so well because that's why transfer learning from color image, black and white, we just make 3 channels the same, made it colored. And this is human data. We go to Macaque. And this is very high level, as you said, classification data. And we did do some post-processing with regression, but this is single neuron data. In terms of scale, it's very different. But amazingly, it beats slightly our own homemade model. So that's, for me, it says that the first 2 layers or the 3 layers will also somehow capture something about the primate brain. That, to me, becomes some evidence. Of course, it's not the proof, but I think we're going to have accumulation of evidence like that to say I was really, really surprised. Because we work very, very hard to use the data we have. And this has nothing to do with our input data. We just took it as black box. This data was collected 10 years ago, right? This is much more recent. And then it became very good processes. And people also report that on radiology, right? They try to use deep learning to replace radiologists' reading of different medical images, and they start the same way. We start with something like this and do some alteration. So if this deep net built on ImageNet captures something, then it's much better to start from there than from nowhere with very limited data.
Bin, can I ask a question?
Yes.
So could you clarify what would have been a negative result in this case? So I'm just wondering, like, you can predict the V4 activity from whatever information comes through the bottom layers, the early layers of the
right.
CNN. But maybe could it be possible that information is just rich enough? It couldn't maybe it's just rich enough to predict V4, but does that necessarily mean that it's somehow similarly processing the data as the brain would be?
I would say that you never know for sure, right? But more and more such applications give me always adding evidence that it's a very reasonable hypothesis. I would say it's consistent. That's why I say support. Doesn't prove, right. But support that it might be capturing something.
Maybe if you could then show that the information in the early layers is not good for predicting some things that the V4 doesn't, or somehow sort of show that it has also similar blind spots as the brain does, then that would be even stronger evidence that it's doing something similar, not just doing rich.
I agree, but we don't have the data right now that's again the data for us.
I actually heard Jim DiCarlo give a talk just yesterday at another meeting at New York. And they had a PNAS paper, which was the one you alluded to. And what they said was that if you could take V4 data and try to see if you can predict it with a linear predictor from V1 from the first layer, the V1 layer, which is the first one that actually resembles the properties. There, you can actually see the receptive fields are identical to what you see in V1, right? In these deep learning networks, they're Gabor functions. They're oriented just like human weasel. So you can see how well can you predict the V4 activity? Not very well. It's just not nearly as good as the 2 or 3 layers up. And then you can also ask the same thing about the top layers, where we know that there are neurons in the infratemporal cortex that actually are very specific to specific objects like faces, right? Can you predict V4 activity from that? And it turns out that not as well as the middle layer, right? So that goes part way toward telling us that that there is some progression in that hierarchy that is somehow recapitulating what might be happening in the brain.
So for us, it's actually we end up with this stimuli we can feed back into the electrode and to prove. That's where I feel like that's would be strong. But in these high dimensions, I agree with you that there are many paths you can do well with prediction. Right, that's why you need other evidence. But usually we start from a good prediction and to do other things. We suggest you do intervention. And we come up with these models with stimuli we think will promote, will get a neuron excited, and we can feed it back and prove that. But still, I mean, in all these games, regression, there's many paths in high dimension for prediction. But it's the structure. So you can try to interpret, and then you need to other physiology or whatever modern neuron data to collaborate these two together.
Right. So I was just thinking that what I meant by blind spots really is that we've seen that neural networks have these vulnerabilities as to sort of small perturbations that we wouldn't really see, just adding some kind of pixels carefully adjusted, and it suddenly thinks it's a completely different object, and the brain doesn't do that. So if you could show that, that kind of properties hold,
you can say that
stronger indication of.
You're suggesting that if our pipeline is more stable to this adversary example, that's good suggestion. We haven't done that. Yeah. So this is just 3 models with AlexNet, VGG, and GoogleNet, and the colored 3 models, and then dotted lines are the real data. So you can see that we have 3 models basically do the same prediction. So quickly, I can use layer 3, layer 4, and that with 18 models. They all give very similar predictions. So that's a modern problem. Even I have classical data, but this is a modern problem because it's very easy, TensorFlow or Torch, whatever, they made it for us. We fix the coefficients and we do regression. It's very easy. Our lab on limited GPU could do that. So which one are you going to take for interpretation? So we decided to go for stability principle. And this is a way to prevent from model hacking. People have heard about p-hacking, right? You have many, many p-value, choose a small one. Same thing, you have many, many models, it's the same thing. I call that model hacking. So this is a way to stay away from model hacking. Sometimes you end up with nothing, but at least you don't find anything wrong. I think science is very important. We control false discoveries. It's much, much worse to have a lot of false discoveries because it's making the building up on each other much, much harder. So we have to really, really control false discoveries, which we not. We have many papers which cannot be replicated. Then we decide to go take this deep dream paradigm again from deep learning. We actually don't do exactly the same. We have now for each neuron, we have a one-dimensional response with input as images. And we just work around and to make it maximize, that's called deep dream, and we actually maximize a regularized version of that. That's what I call deep tune. And these are the 2 images from Lasso model and Ridge model after AlexNet. They have very similar cross-correlation performance on test set, 0.63 and 0.64. And they both give this curvature kind of indication. Right.
So these are images which if you give it to the monkey would have maximally stimulated that neuron.
The model neuron.
The model neuron.
Exactly.
Yeah.
So that's why we say this characterized neuron, and they look very different from, remember, all the very synthetic complex shapes, right? This is a lot more natural looking. And I'm not going to interpret these little things because they are not consistent. So I use stability, only the big picture. But it's possible that the neuron get equally excited with both of them. We don't know. Neuron doesn't have to excite only about one particular stimuli. It could be a whole population. So actually give you a whole population. And this is to show you that the convergence is pretty stable. So you start with white noise right there, and then it converges to that. And this is a way to see it with paper without the dynamic movie showing. It's called superheat, to look at high dimensional data. I have 18 models. Here's 9 of them. This is layer 2 regression, layer 3 regression, layer 4 regression. This is AlexNet, this is VGG, this is GoogleNet. You can see they all have their own. GoogleNet have this eye-like looking things. Again, I'm not going to interpret, but the same curvature-like things appear. So I can put this back if the electrode is still in place with computation and prove it to see which one maybe the neuron likes better and to differentiate it. But maybe liked all of them. Right? It's possible if one neuron likes all of them because they only concentrate on these things that matter to them. So this becomes experimental design. And with high computing power, we can do that.
Actually, the edges all on the same place, so if you would superimpose them,
they're not exactly on the same place. We try to quantify, actually that's not easy. But V4 also has invariance, right? So it's hard to give a quantitative measure. We tried, it's just they're quite different. Yeah, but visually to us, very simple. Maybe impose.
So these artificial neurons, do they have a receptive field, or this is the entire image?
Yeah, I tried. The receptive field, we think is more like, this is where the tuning region, smaller. Yeah, it's not the entire image. You're right, we could have do that. This is the 2 for this and that. So it's more this region. Yeah. For longer talk, I have that. Yeah, I can show you after the talk.
Can you say just a few words?
Yes.
Can you say a few words about, I mean, it looks like GoogleNet is quite different,
Yeah.
at least visually, from the other 2.
GoogleNet is the largest.
It's the largest one,
Yeah.
right? So what layers are these? How many layers in?
We took the same layer 2 from the different nets.
It's layer 2?
Yeah. This is layer 3, this is layer 4.
I see. But layer 2 is already, how many convolutional layers is layer 2?
How many?
How many total in the whole network?
How many? I think we only look at, I think GoogleNet is like 9. More than that, 13 or something?
AlexNet is only 5 convolutional layers, and GoogleNet has much more than that. And layer 2, 3, 4 is what we mean.
Yeah, we only took the early ones.
The second convolution layer, after the second convolution layer, and after the third convolution layer, and after the fourth convolution layer.
Yeah, so this is more counting as convolution layers.
I see. And can you also just say something, I know that this is not about computability, but can you also say something about how long it took to do each of these 3 analyses?
But for this, we took already pre-trained, so it's pretty fast, right? We only have to do a regression.
I see.
Yeah, so this is nothing, right? It's not very expensive. So if we retrain it, we didn't, right? We could do that, go back, but we haven't done that. So that's the thing, we took the fixed box and used physics extraction. So I thought that was more powerful to show the power of the deep nets than retrain it. So for each, if you start with the same initialization, we have 18 different such images. And we can also try to have a consensus. We just say for each starting point image, we have 18 gradient because we have 18 surfaces. We're going to choose the direction that's the slowest changing and take the sign and move that direction. So we have a combined, very conservative way to produce these images, and you can see that's what we got. So we try to quantify, to unify them and say, oh, let's human look at it, look similar. And we also did compression, removed 80%, 60% of the nodes, and then we see the pattern remains. So we did a lot of alteration to show that this pattern is real. Unless you go to, if you remove 95%, things get a little screwed up. But 80% is pretty safe to see the pattern still there. And we also went back to the original image database to see what are the images that excite this model neuron. And you see curvature again in these images that excite the model neuron. So we just try to have multiple sources of confirmation that this neuron cares about curvature. Use the same methodology, we have 10 of them. So now you see the diversity of different neuron patterns. And it's very different from what you saw with the synthetic complex stimuli. Right? This looks a lot more natural. And this can be proven, but Jack doesn't do physiology anymore, so that we can feed back. I've talked to James at the NIPS workshop last year, but we haven't really connected yet to see whether his lab can do some of the follow-up to prove it. Yes?
So has anyone ever tried to do like finite difference method differentiation to backpropagate and do a deep dream style on a real actual neuron?
I don't know. We didn't do much the deep net training at all, right? We took them, but not. I'm aware of the people do back propagation. In some sense, I don't think the back propagation will be that different if you don't do the derivative, you do the difference. But computationally,
I'm just thinking that this process, I mean, so there's a whole literature about getting sort of maximal stimuli out of these neurons.
Okay.
But it just strikes me that one could actually try some kind of evolutionary approach or finite difference approach to get the image.
Okay, so you have to understand the limitations of having a monkey. You're very limited in terms of the amount of time you have.
Even if you did it for one neuron, I'm sure you get the hundreds of.
So the Connor results that the Pasupathy and Connor results,
yeah.
they were, as I understand it, they're actually they're a kind of descent optimization. So they actually show succession of images. It's actually a gradient, a genetic algorithm. So these throw a number of different shapes, and then whichever one is the most preferred by the neuron is then used to generate descendants that have similar properties, and then they choose the maximal. So they're trying to optimize. So there is an algorithmic approach behind that.
But that they've constrained themselves with the kind of the synthetic stimuli.
Yeah. Well, they have some feature space that they're trying to progressively search, and then they'll seed it from different start points to see if they end up in the same.
So we didn't constrain in the image space. We constrained in the optimization space. We made it. You only work on a smooth surface. So that's how we constrained. So high level, I see very similar, but specific algorithm. Yancy, you want to add?
Having a model and not having a model, there's a huge difference. So if you want to do it directly on the real neuron, you don't have model. So search space, like imagine you have a 200 by 200 pixel search space. To do that, search space is too large, like if you want without model doing the search in that space. But if you have model, like here you have a model of this neuron and you have this gradient, like using gradient method to do the search will be more efficient in this image space. So that's why, like, I think doing this in real neuron will be a hard task to do.
But I think there can be a combination, right? So we can use it to search and feed back to see the real neuron.
Yeah, I'm just noticing that, so in the sort of latest deep learning research, lots and lots of people are using so-called evolutionary strategies to get gradients for functions that are much more complicated. And it just struck me that maybe one should revisit that approach in a monkey brain for getting an optimal stimulus. That's sort of the...
Yeah, but I think, as Yancy pointed out correctly, that we can develop some initial, probably study an initial model and use it guided too, so you don't have the whole space. So that would be a nice combination.
Have you looked at these in any sense orthogonal to one another?
We have another plot. A lot of the neurons, they're not always orthogonal. Some neurons actually like similar stuff. There's another plot I'm not showing you. You can group them a little bit. We feed these preferred stimuli to different neurons, and they really prefer themselves, the deep tune from their own model. Yes?
So I have a problem actually really understanding what I'm seeing. So you said it's like a preferred stimulus, maybe in the sense that it maximizes the neurons, say, firing rate?
Yeah. The model.
Generally,
the model.
Right?
Yeah.
But generally, if I think about simple cells, preferred stimulus is defined under a power constraint in some sense, right? You would say, I mean, in some sense, you have a simple cell, for instance, right? There's a big image space, and it's a mapping into a single dimension in some sense, which is the firing rate of the neuron. Yeah. So there are many stimuli that will elicit exactly the same firing rate. So to find the preferred stimulus, you'd have a power constraint that says, okay, I look, say, on a hypersphere, basically what is the stimulus that gives me the maximum firing rate. So is there a similar power constraint here?
We did a constraint on the loss function. So we could add the power constraint in the search space, which we didn't. We kind of get the smoothness through. We have a regularized version of the model. That's how we did it. It's a little easier computationally, but we can talk to see. We're not very familiar with the power constraint. We'll be happy to talk to see whether we can constrain and what's a reasonable constraint for V4. For V1, I think maybe there's more agreed upon constraint. For V4, for V1 maybe, but for V4, do we have a good way to constrain the space of the images?
I don't.
Yeah, I don't. I'm not aware of. Yeah.
So on a related point, it's not clear to me at all that a single neuron should have a single preferred stimulus, right? Because, again, it should lie on some very high dimensional...
So we have a population.
It should be a whole distribution.
But she said that if you start from different initial starting points, you get different images, but they all have the same qualitative look, right?
Yeah, exactly. So we see that you can characterize, you remember this one? Right. We have, that's just my initialization from 8 to 9 models. I can have many, say, with little alterations, changes from, so I really have a population.
It would be really interesting to know what that distribution, how it's related, right? Like what are the invariances?
It hasn't been easy to characterize. Yeah, we tried. It's just quantitative to say whether it hasn't been easy. So modeling that actually become another statistical problem. But we think we want to see at least somebody doing some experiment before we do more.
But it's interesting that your human visual system can see a similarity, right?
Yeah.
And so you're right. Quantifying that is very difficult, but that's the problem of vision. It's exactly the problem that we're trying to solve.
Yeah, I gave you the IBL, and the physicists look at that. The spacing seems to be constant, which only physicists told me that. All the other didn't, right? So everybody see different things with what they're used to. And if you look at the spacing is kind of the same, but how do you find that? That's not trivial. We tried a little bit, we didn't succeed.
How do you know that's not just an artifact of the GPNet?
That's why we need to feed it back. This one have good prediction performance, right? And also in the literature, people know we found neuron care about curvature. These are all the evidence. And we also have similar things from earlier model to very different route. Right, so that's also the thing. We have two-layer sparse coding models, which is completely different, not deep nets. And we have similar results. So that's why exactly the stability. That's why eventually the proof is feed it back for the single neuron to get a single neuron to fire. But the prediction performance is real. As I said, in high dimensional prediction problem, there are many equally good prediction models. I don't know for sure. Definitely. That's the challenge to go from prediction to interpretation. That's always the problem. And this is using the consensus deep tune. You can see a little weaker signal, but this shows something. This 18 model have some consistency. Through this, we can show some consistency. If we define this by the metric, we have some weak metric that there's consistency. We take the weakest, like the lowest gradient, and take the sign, we get something very similar. This is over 18 models. Right? So all of this, we've been beating ourselves to ask these questions, and we're ready to actually write it up for the rest of the community to judge. Okay, so now that's really classical data to have the kind of state of our machine learning to bear and deal with interpretability and try to see how we can prove it. Yes?
So this may be a discussion question, but it seems like you're transitioning, so I'll ask it. I'm deeply sympathetic to the idea of interpretability, but I wonder, what does it mean actually? So you've shown sort of maximal stimuli that maximally stimulate particular neurons in these artificial networks.
Yeah.
So you have, for example, small models where interpretability might be easy to understand, a linear approximation, a small norm solution, or low rank, any of these things, right? But now you get into a large system, and all of a sudden it's not even clear to me that the image that maximally stimulates a particular neuron is potentially relevant to what the system is even doing.
It's a way to describe the model. Actually, my group is writing a paper exactly try to define a few things in interpretability. We define as domain interpretability is like to this audience that what I say makes sense. So you need to have the audience in to really answer the question, what's interpretability? And there's a lot of things we call algorithmic interpretability. It's like the mathematical form where it's simple enough for people to take a look. So we separate the domain interpretability with algorithmic interpretability.
But save the question, it's a really important one, because really what's going on now is, I think, the real key issue, which is how do you deal with models in spaces that have millions and billions of dimensions, and how do you characterize distributed representations? So with any given image, you find activity patterns throughout the entire network. It's the entire network that's recognizing the image. It's no single neuron. And so you have to understand something about joint statistics if you really want to answer your question.
And also, yeah, interpretability to whom, right? Is that human in the loop or human not in the loop? Is that just compression? So there's a lot of, I think we need to design some concept that the community can really understand in the same way, and then we can discuss to the next step. Otherwise, we're all talking about different things. So this is a little transition, I think that I probably have a little bit of time. That is the paper by Friedrich et al. From Liam's group really deal with high throughput image data for large population neurons and image data. So I want to share something not for neuroscience exactly, but image data, they use quite heavily constrained non-negative matrix factorization, see how stability can come to help to select the number of components. For them, I think each component corresponds to a particular neuron activity. And for us, it's actually development biology, it's about a pre-organ region. But there's a lot of information of the experience we can leverage across different fields. I think that's really one thing to be aware for this community is we don't have to reinvent the wheel every time. It's very inefficient. So in that spirit, I will share this very quickly. So this is a project with Urban Fries from IBL. The lead student is Siqi Wu, and it's appeared 2 years ago, PNAS paper. So this is trying to figure out how Drosophila develop mapping of cells. The different parts in the embryo stage of free fly develop different organs for the adult fly. So this is pre-organ region, that stage that morphology doesn't tell you what's going on yet, but genetics already there. So this is, we have many of these spatial gene expression. So this could be the brain, right, the fly brain. And then you have, actually there's such a data for Genela farm that you have different genes where they're expressed in the fly brain. We just haven't used that data. There's a lot of registration problems. And here we use this called fate map from Drosophila. You know, Drosophila is a star in biology, has won 6 Nobel Prizes. That to figure out
the
this is.
Drosophila won a Nobel Prize?
What?
Did you say that the Drosophila won the Nobel Prize?
Well, the people who work on Drosophila won the Nobel Prize 6 times. To be precise, it's a star. Yeah. So they sacrifice themselves. So this is the region saying that it's a neuron region. So this is really people figure out with old technology. We want to use high throughput. And genes interact locally. They don't just do the same thing. This is the area that the different genes team up in different ways to form build different rooms. So giant is a gene at different locations. They interact with different networks. Okay. So after tons of work, that's another thing. It's pre-processing of data. We have to give people prizes for doing that. The modeling is actually a relatively small part of the hard work. It's registration, especially with high throughput data. If you don't process it right, you're pretty screwed. So here we end up putting them in ellipsoid, and tons of work also using other people's software. This is public data. And then we downsize it into 4.5 or 405 pixels for 2 reasons. One is for computation constraint. We couldn't deal with it otherwise. The other is after we put them into the ellipsoids, they've been stretched. We cannot have very small pixels. They won't match. So you have to have big pixels so that they will correspond to each other. Because the registration. We end up, actually we borrowed from sparse coding. That's the first time I got this project. I happen to be doing neuroscience. This is a later project than my collaboration with Jack. We use sparse coding now, we move on negative matrix factorization. So we lay them out. So this could be different measurements of the high throughput neuron population data, and you end up with different neurons at different locations. So for us, it's different organs, right? And I think in that paper, they used constraint. We didn't use constraint, but I think what we say here is very similar. So you basically use positivity as a sparsity constraint to decompose these different genes into different, we call principal patterns that correspond to the different neurons in the neuroscience world. And amazingly, we're pleased to find that the regions become all coherent. We didn't put any continuity constraint. They came out to be like coherent regions, which makes sense. This is like pre-organ regions for development biology. And what stability brought to the game is to find how many principal patterns. It's like, why I should have 21 column, 21 regions, or for you guys, how many neurons there are. And we use a stability criterion to say that you start from different random starting point. This is a non-convex optimization problem. And you match them to find the best match through correlation. And then you try to say that uses the 1 minus the best match of correlation as a dissimilarity metric. You want to minimize this dissimilarity, and that's how you choose the number of components. So this can be borrowed over to choose the number of neurons in your processing. And we did a lot of analysis for 22 for the same stability principle. This is awfully close. The data is not very clear. So we also did a lot of analysis saying that what's going to happen here. We don't want to have a completely different signs because we take 21, not 22. So again, stability.
Why not just do PCA or something?
What?
Why not just do PCA?
Oh, PCA give you checkerboard like a high frequency doesn't make biological sense. We did that. It's because it's all positive. It's the constraint is the wrong constraint. The variability is the wrong constraint. You get these patterns, biologically doesn't make sense.
So yeah, PCA, a couple of things. So PCA is trying to, it's really reduction. You know, trying to get the maximum amount of the variance into the minimum number of components, which means it will pack together things that are really unrelated. And if you want to separate out what are the independent components that are positive, then it turns out that, and also these algorithms are much better. But actually, PCA is, if you have Gaussian data, again, at the beginning, if it's Gaussian data, PCA is the best you can do, it turns out. But all of the data here are non-Gaussian.
And they're sparse.
And in that case, the ICA, independent component analysis, and non-negative matrix factorization have been shown in many, many data sets to give you where the joints are in the data.
Yeah.
And the independent things here turn out to be little neurons or regions which are spatially localized, and it didn't have to be that way.
Yeah.
So can you explain this graph a little bit more? You're talking about the stability of the number that it picks, right? Which also has
No, so what it did, I have a problem of choosing how many components I want, right? This is I lifted that 21 is a 19. Right. So what I did is I fixed the number of components, and I do different random runs. Right. And then I match to have a measure how the different local minimum, the principal patterns are similar. So I want them to be as similar as possible.
Because like with non-negative matrix factorization, a lot of times when you increase the number, it changes all of the basis functions, right?
Yeah. Yeah, so we choose one that give you the most similarity.
Of the actual patterns themselves.
Use correlation as similarity measure, yeah. So this 22 actually split one of the 21 into 2, and we used biology to decide that was not desirable. So that's the patterns we get, the principal patterns. And then we can do some biology on it. We can localize the genes and do local networks and all that. So there's a huge amount of work followed. This is so the question, if this is shaky, then the rest just go down the drain, right? So that's why we work very hard to say, how can we trust these principal patterns? It came out from a magical mathematical algorithm, and there are options, right, to do this way and that way. So one thing is important is we need replication images. This is also a lesson, I think, in neuroscience. If you can do replication. And also, we also use existing knowledge from this Drosophila to really try to give functionalities to computer genes. Basically, you can localize it. So for you, it's like you have different neurons, you use that. And also, we use prediction, human labeling, and a particular known gene network. And we did theory, too. We end up using sparse dictionary learning, stochastic models, to analyze one of the algorithms. So my view on theory is that you should have multiple models, very generative models, and to make sure your algorithm works well under multiple models. That's more robustness. Unless you know one sense, really, that's the right one. Otherwise, you want robustness. And also knockout experiments. So people actually did a CRISPR-Cas9 and try to discover genes. So that's similar to feed the stimuli back to the electrode. So to go to the neuroscience, I see replication is important. And you need some reality check. I don't know the data well enough, but prediction is one possibility. And you need to know all of the stabilities relative to your data perturbation, your algorithms, and ad hoc human decisions. Your lot of hyperparameters you just chose. Would that be driving force, right? Maybe it's not for one lab, but collectively we have to really do all this stability analysis. And do you have new discoveries of functionality for unknown brain regions? That's one thing you can see. And prediction performance, if people can label some data, can you do well in prediction? And also reconstruction, we use a particular network, a GAPT network. Do you connect on data? Can you connect with that, do some prediction with that? And theory, I think, but you want to do analysis under multiple models so that you have the stability. And also, can you do intervention to feed it back?
So I have a little anecdote about that.
Yeah.
So when I first moved to the Salk Institute, Francis Crick was there, who is a god in biology, discoverer of DNA with Jim Watson. And so he used to come to tea almost every day in my lab. And I remember once he said, Terry, you're very fond of your models, right? And he was right, but that's what I do. That's my business. So we all perhaps are a little bit too attached to the approaches that we use. Okay, I have to admit to that. He said, but what you don't understand is that the purpose of a model is to design a killer experiment that nobody else would have ever thought of doing that's going to give the game away, right? And that actually goes beyond prediction. Prediction is good. It tells you on the right track. But unless you have, the model leads you to a new framework, and you can test it, that's when you actually have a discovery and the potential for a deeper understanding. And I think that's what we're looking for here, right? We don't have it yet, but I think we have some of the tools that will take us there.
Yes, a lot of these are called scientific recommendation system, right? You come up with hypothesis or something, and you go back to the lab to the intervention. That's finally the proof. But a lot of time, even in genetics, it's relatively simple. So you have to bridge the gap using some theory, and maybe later technology become available to do more complex experiments. So there's some theory related to three I won't mention for this, but I just wanted there's a theory arm to this, and it's just a summary that for the PCS workflow, I think stability is extremely important, and many different steps. Prediction people already bought. And I think for the modeling, our experience that you need some matching, structure matching of the domain knowledge to build into your data, even with kind of data rich. But relative to the complexity of the problem, we're kind of data poor. Thank you.
So any burning questions now? We have a good problem here. We are a little bit behind schedule. But there will be a discussion session after Marian's talk. So then I would say let's go into the coffee break. It will be 20 minutes, and you will hear the bell ring to call you back to the room.
Thank you. That was